Thank you. And thank you to the organizer. Oh, sorry, there's an echo from my computer. I should turn down the sound. Sorry. So, thank you for the organizers for inviting me, and apologies for the audience for my not being able to come in person due to obligations at the university. So thank you for giving me the opportunity to give this presentation online like this. The aim of my talk is to present to you the work we have done during the past 4 years and to hopefully convince you that lymphoblastoid cell lines are a good tool when searching for biomarkers, at least as a preliminary tool before you collect patient cohorts. And please keep in mind the work that I will present to you was done with lymphoblastoid cell lines from healthy individuals. So of course everything needs to be corroborated in clinical cohorts. So what I present to you are preliminary studies and we have some tentative biomarkers which I hope that others will help us to validate in clinical samples. So the study is about depression, and I'm trying to move forward. When we did the test before 1 hour, it worked perfectly, but now, oh, okay, it took some time, sorry. So, we all know that there is a strong need to find biomarkers for treatment response in depression because there are several drug classes to look for. We have the SSRIs, the SNRIs, the tricyclics, serotonin ligands, and finally lithium augmentation therapy. All those therapeutics can be used to treat depression. However, the response rate is not good. It's at best about 2/3, with over 1/3 of patients not responding to first-line treatment, which is in most clinical centers, the SSRI antidepressants. And moreover, it takes at least 3 or 4 weeks to see if there is a clinical response, and meanwhile, there's suffering for the patient, for families, and of course, increase the risk of suicide. So if we had biomarkers which would tell us which is the best drug class for each individual patient, this will be a great achievement in psychiatry. So the first pharmacogenetic study on depression was published 15 years ago in molecular psychiatry by an Italian group that showed that the polymorphism in the promoter of the serotonin transporter, which is known to be the drug target for SSRI drugs, which bind to it and inhibit the reuptake of serotonin. So they showed that this polymorphism is associated with treatment response, with the longer promoter allele associated with better clinical response. So this is one of the best-known studies in psychiatry, I think, and it was cited hundreds of times. There were thousands of papers about the serotonin transporter polymorphism, many of them initially showing that indeed it can be a biomarker for treatment response, but eventually in the last 2 years, there were 2 large meta-analysis studies published with the conclusion that there is no clear association between the promoter polymorphism of serotonin transporter and treatment response to SSRI drugs. So obviously this was a classical example of publication bias. Everyone who could reproduce it published initially, and it took many years for others to publish the negative results. But now we have the conclusion that this polymorphism, which is very famous, cannot be a biomarker for choosing a treatment for depression. So obviously people continued with GWAS, with genome-wide association studies. The first studies were published 3 years ago in 2010, 2 very large studies, including the study trial and the European trial, each of them with hundreds of patients treated with SSRIs. 2 more studies were published subsequently, but very quickly it became clear that this is not good enough because there was nothing in agreement between these different studies. So the Mars gender-dependent study, each of them reported different SNPs as associated with the response to antidepressants. None of them had very good p-values, and none of them were in agreement between the different studies. So the conclusion of a recent analysis and review that was published about this is that from GWAS, we cannot come to find biomarkers for treatment choice for major depression. And this is not very surprising because similar obstacles were encountered in other areas of genomic research, including cancer. And I would like to turn your attention to this beautiful commentary by David Goldstein that was published already in 2009 in the New England Journal of Medicine about missing heritability, in which he claims, rightfully so, that common SNPs represent a tiny fraction of the human genomic polymorphism and are not a good tool to study pharmacogenetics. So obviously the next step is to do sequencing. And we were aware of all this when we started this project 4 years ago, but sequencing at that time was still prohibitively expensive. It still is rather expensive, and we opted for another method, which I will present to you, and this is using the collection of lymphoblastoid cell lines, which we have in our lab anyway, because we are the National Laboratory for the Genetics of Israeli Population. We have a collection of about 2,000 lymphoblastoid cell lines, and these cell lines come from healthy donors that represent the variation of the human genome. And we realize that we can use it as a tool if we define in vitro sensitivity to SSRIs as a surrogate for the clinical response. So we did not work with clinical samples, but with lymphoblastoid cell lines that are immortalized lymphocytes from healthy donors. And we looked at an in vitro response of growth inhibition by paroxetine, one of the SSRIs. And we chose paroxetine because it has the highest affinity for the paroxetine, for the serotonin transporter. And it was reported to inhibit the growth of many cell types, including cancer cells. And in fact, SSRIs were suggested as add-on therapy for certain kinds of cancer, like lymphomas and gliomas, which never happened for obvious reasons. Drug companies don't like to change drug labeling for such good money makers. But anyway, this was in the literature when we knew that SSRIs inhibit growth inhibition. So we chose to use them to phenotype different donors, cell lines from different donors, look for donors whose cell lines are very sensitive or very insensitive to growth inhibition by paroxetine, and ask the question, what is different in the expression profiles, in the RNA of these cell lines, and maybe come across a gene that its expression level can serve as a biomarker for treatment response. And this is what I will present to you today. You have to bear in mind that lymphoblastoid cell lines present the exact same DNA sequence as the donor. The immortalization process with Epstein-Barr virus does not change the DNA. The virus stays dormant in the cytoplasm and the DNA is intact. So nuclear DNA is not changed, mitochondrial DNA is not changed. The epigenome is changed a bit. You can see here there was a study reporting that there's only 85% matching in the epigenome, in the methylome profile of lymphoblastoid cell line and lymphocytes from the same donor. So it's not a perfect match with lymphocytes of the donor because the epigenome is changing a bit, but the sequence is exactly similar. So the approach is to look at the phenotype of the cell lines, ask what is different between very high and low sensitivity cell lines, and then look which genes or microRNAs are involved and try to study these genes and understand how this is related to the mode of action of antidepressants. It is very important first to remember that both lymphocytes and lymphoblastoid cell lines, for short LCL, express functional serotonin transporter. We have shown this 9 years ago, and this is an important point to remember. If you want to use lymphoblastoid cell lines for a study on CNS drugs, you first must make sure that the drug target is expressed. So for SSRI, this is a good tool because the serotonin transporter is functional and expressed. For some drugs, it's not functional. For example, we tried Ritalin, and it did not work. Ritalin does not inhibit or does not have any effect on these cells because the dopamine transporter, the drug target of Ritalin, is not expressed. But for SSRIs, it works beautifully. I hope to convince you with the next slides. So we did the phenotyping experiments, which you can see everything is published, so I will go very quickly, but just you can see in this slide that there is a large range of sensitivity to paroxetine. some cell lines on the left are very sensitive, some cell lines on the right are relatively insensitive. I also want to show you that there is correlation between different SSRIs. So if you measure sensitivity to paroxetine and citalopram and compare, you see nice correlation. If you compare paroxetine and fluvoxamine, again you see very nice correlation. So it means that what you measure, the growth inhibition phenotype, is related to the major drug target, to the SSRI, to the serotonin transporter that the SSRIs bind to, and not to some off-target, because these are, after all, different drugs with different chemistry, and some of them have their own off-targets. So this is related to the major target. Just one more example in the next slide. You can see that beclomethasone and dexamethasone correlate beautifully. All this is published already 3 years ago. But if you compare paroxetine and beclomethasone, there's no correlation. So this is another proof, okay, that what we measure, the phenotype that we measure, is relevant for the mode of action of SSRIs for inhibition of the serotonin transporter. So next step was to choose the edge cell lines. We chose only 7 from each edge. So altogether we did 14 microarrays, a very small number, I admit, but that's the budget we had at the time. And we asked what is different in the entire transcriptome. And according to bioinformatics tools that were used by the bioinformatics team at our university, there were 61 genes showing differential expression. We know for statistical analysis that about one half of them, about 30, are probably false hits. So we are not sure of all those 61 genes. But the top one, CHL1, we are very certain of it, and I will show you why. It's important to know that none of those 61 genes were related to serotonin, not the transporter or receptors or synthesizing or catabolizing enzymes of serotonin. Nothing was related to serotonin. What is known about CHL1? CHL1 stands for close homologue of L1. Both CHL1 and L1 are cell adhesion molecules that function in many tissues, not only in the brain. But what we found was 6.3-fold lower expression of CHL1 in cell lines that showed higher sensitivity. So there was less CHL1 in cells that expressed very high sensitivity to paroxetine. This was confirmed by real-time PCR that showed even a bigger difference because there is often data compression on microarrays. So we looked at papers about CHL1. It was already reported that mutations in CHL1 were implicated in certain cases of schizophrenia and autism, but very rare cases because there are about 100 genes already linked to schizophrenia and to autism. More importantly, it is known that CHL1 is involved in neuronal guidance, and I will show you the most important study in my mind that connects it to our finding. When this group that published 2 years ago the work did double knockout mice, they knocked down both CHL1 and L1, which is very close to it, it's very similar. And what they observed in these mice was a posterior shift of axons going from the thalamus. Instead of going to the prefrontal cortex as they normally should during embryonal development, these axons went to the visual cortex, so they went more posterior to the visual cortex. This means that for the formation of correct limbic system, you need L1 and CHL1. So we found a gene that is function in the guidance of neurons from the thalamus to the prefrontal cortex. And these, as you know, are serotonergic neurons that are involved in mood regulation. So this showed us that very likely we found a true hit. We found a gene whose level of expression are relevant for the in vitro response to antidepressants, and the product of this gene, the cell adhesion protein, functions in the guidance of thalamic neurons to the prefrontal cortex. And soon after we published this work 2 years ago, there was already a first citation from a clinical work, and this came from the study, nonetheless. And this was a report by Clark and colleagues about side effects of citalopram. In this fact, they reported dizziness, and they found 5 SNPs in their SNP analysis, genome-wide, 5 SNPs that are correlated with dizziness associated with citalopram, and one of them was CHL1. So CHL1 is somehow related to the mode of action of SSRIs. It is true that here the report is about an adverse event, but keep in mind that adverse events sometimes go together with efficacy, because if an individual is more sensitive to the efficacy of the drug, it could also be that the same individual will be more likely to get an adverse event because everything that is activated by this drug is happening more easily. So this I think, is a nice partial confirmation that CHL1 is related to our finding. Now I shift gears and talk a little bit about microRNA, and we'll show you what we did. So I believe most of you know, but just for the few people who don't remember, microRNAs regulate gene expression by binding to the 3' UTR region of the gene and destabilizing it and leading it to degradation. So microRNA destabilize genes and this way control the level of expression of RNA. And it's important to remember that we have only about 1,000 microRNAs in our genome compared with about 25,000 genes. So it's easier to find a needle in the haystack, so to say, when you look at microRNA. And this was the next step of our work. We did a similar genome-wide expression study for microRNA with the same cell lines, again comparing the very high and low sensitivities to paroxetine. And indeed, with bioinformatics tool, we had a list. There were several microRNAs that we found, but the top one, the one showing the highest difference, I admit not a very good p-value, but remember it's a small sample. So the top one was not so known microRNA. It's called 151-3P. But what was for us fantastic is that this microRNA was reported by bioinformatics tools to target CHL1, which shows the opposite direction. So near 151-3P shows higher expression in the sensitive cells together with the lower expression of CHL1, and this goes together nicely. So this indicated to us that CHL1 is a true hit because we have a confirmatory finding about the microRNA that regulates it. And here I don't have time to show you, but meanwhile we did experiments that really prove that this microRNA targets CHL1. So it's now not only bioinformatics tools, as we say in the paper, but hopefully in our next paper we will publish and show that this microRNA regulates CHL1. I have a few more data slides to show you that I hope will be interesting for you. First of all, something we published just 2 weeks ago. Sorry, it still says in press. It came out 2 weeks ago. You can see it open in line on Transnational Psychiatry. So we did a different experiment, but again using cell lines from healthy individuals. In this case, we used only 4 cell lines. We were limited by budget considerations. And we asked what is happening to the transcriptome, what changes in the gene expression when we expose cells for 3 weeks to paroxetine. We chose this time of 3-week exposure because this is the time that in the clinic you start to see clinical response. So altogether we had 8 arrays, 4 from control cells exposed to cell line, and 4 from cell lines from healthy individuals exposed to paroxetine for 3 weeks. And now we got a list of only 14 genes showing a genome-wide significance difference. None of them related to serotonin. The serotonin transporter is not changing when you expose for 3 weeks to paroxetine. The receptors are not changing. None of the enzymes relevant for serotonin are not changing. We were also disappointed that CHL1 was not changing. What was changing now? We found another gene, integrin beta-3, ITG beta-3. Integrin beta-3 is also a cell adhesion molecule, a very famous one in cardiology because it's highly expressed in platelets and it participates in platelet aggregation, which is crucial in thrombotic events. It's also known as CD61 or GP3A. And this gene showed almost 2-fold higher expression upon the chronic treatment with paroxetine, with very good p-value this time. So what do we know about integrin beta-3 in the context of neurology? It is expressed in the brain, in particular in the hippocampus and frontal cortex. And most importantly, it is essential for the serotonin transporter activity. This was reported about 5 years ago by a group from Vanderbilt University who showed that in knockout mice for integrin beta-3, you lose the serotonin transporter activity. So this gene is a cofactor. It codes for a cofactor of the drug target of SSRIs. This gene is also implicated in autism. Some cases of autism are related to mutations in integrin beta-3. And it was very recently reported to be related to age of onset in schizophrenia in some cases. So it was not directly, mutations are not linked to schizophrenia itself, but to age of onset in schizophrenia. Moreover, we looked at microRNAs in the same experiment and asked which microRNAs are changing, and here we found 2 microRNAs, MIR221 and MIR222, that are both predicted to target integrin beta-3, and they were downregulated while integrin beta-3 was upregulated. So what we think is happening when we treat with paroxetine, at least in tissue culture, we first have downregulated of these 2 microRNAs, which allows, and it takes time because first you need to get rid of the pool of microRNA that were in the cell. But once they are downregulated, and this takes several weeks, you see the upregulation of integrin beta-3. And this was also with very good p-values for these 2 microRNAs. So I want to get close to conclusion just to present a model how integrin beta-3 could be related to response to SSRIs and how this is connected to CHL1, which I showed you in the first part of the talk. Because there are no studies in PubMed that have both CHL1 and integrin beta-3 in the same paper. I mean, our paper is the first one that puts these 2 genes together in the same study. What we suggest, this is our working hypothesis, we still have to prove it, is that both CHL1 and serotonin transporter compete on a limited pool in the membrane of integrin beta-3. So what happens is that if cells or neurons have a lot of CHL1, then more integrin beta-3 interacts with CHL1 and less integrin beta-3 is available for the serotonin transporter that becomes less active, and if it's less active, it cannot bind SSRI drugs as well, and therefore SSRIs are not efficient in this case. Okay, everything I say so far it's in tissue culture. Of course, we have to prove it in animal models, and this will be the next step of our work to try to show all those things with animal studies. I just want to show very quickly 2 more data slides, and then I will conclude. I think I'm within the time. So I would like to show a study that was published, shown as an abstract very recently in a meeting in Miami by my good colleagues, Alessandro Serretti, Julia Stingel, and their colleagues. Julia is here in the audience and will talk later in this session. So in this study, they looked at clinical samples and found that a SNP, which is intronic, so we do not clearly understand it, but an intronic SNP in CHL1 is related with not very good p-values, but still we had this p-value for relation to non-response and non-remission for the TLL in this SNP, which is in CHL1. So we believe we found something relevant. And regarding this SNP, we did some bioinformatics work, and my postdoc, Elena Milanesi, recently showed that this SNP is related somehow to expression levels of this gene. So we do not understand it because it's an intronic SNP. Maybe it linked to something in the promoter or in the exon and intron boundary and related to alternative splicing. We do not know yet. There's a lot of work waiting ahead to do. But this is probably related to what we found about expression levels and the response in vitro that we can measure. So, as I said, right now we are looking at sequencing of these genes in collaboration with Alessandro Serretti and others, and we plan animal models, including studies with integrin beta-3 knockout mice, to check all those working hypothesis models that integrin beta-3 and CHL1 both play a part in the mode of action of SSRI antidepressants. So to conclude, the message of my talk is, first of all, that lymphoblastoid cell line sensitivity to growth inhibition can be a good tool even for CNS drugs. I should say that I should not take credit for inventing this method. This method was used by several groups for cancer drugs, and it was shown that you can use LCLs to find biomarkers for efficacy or side effects of cancer drugs. There are many good studies, at least 20, about cancer drugs. But our group was the first, and for this I take the credit, that used LCLs for a CNS drug, in this case paroxetine, and we believe that LCLs are a good tool also to look at also to look at CNS drugs. We now have projects with antipsychotics and mood stabilizers such as lithium and valproic acid to look and try to find biomarkers genes using the same tool of lymphoblastoid cell line. And lastly, I want to thank my colleagues. First of all, everything that I described here was done in collaboration with my good colleague Noam Shomron from our faculty, and with our graduate students, Ayelet Marag and Karen Oved, and with help of the bioinformatics team of Tel Aviv University, Varda and Metzada, and our new postdoc, Elena Milanesi, and collaboration through Aaron Neuron with my colleagues in Italy, Alessandro Suretti, in Germany, Julia Stingel, in Krakow, Poland, Andrea Pilk, and also work that I did with Mark Van Sant in Paris. And I would like to conclude here again with a nice painting by Van Gogh. This is the recently discovered lost painting of Van Gogh. And thank you for your attention. And now I can take questions. Thank you very much.
Well, thank you, David, for sharing your time with you. And can you hear me? David, can you hear me? So perhaps no. Thank you for sharing your time with us and giving us this presentation via this way. I hope everyone could understand your talk. It was not